Hello, my name is Xiaotong and I'm from the University of Queensland and Baker Heart and Diabetics Institute. This project explores the essentiality and biological significance of glycosylation and the use of deep learning to enhance unlinked glycosylation site predictions and variant pathogenicity assessment. Glycosylation plays a critical role in protein stability, trafficking, and function. However, most existing prediction models rely on short sequence windows and lack structural context, limiting their accuracy and biological relevance. To address this, we developed SGL-GLY, a deep learning model that combines full-length protein sequence embeddings from probird with structural features. This study had two key goals. First is to evaluate the essentiality of glycosylation by analyzing the distribution and pathogenicity of variants at glycosylated residues in the general population. Second, to build an accurate unlinked glycosylation prediction model using both whole protein sequence and structural context. So first, we curated a data set of protein with annotated unlinked glycosylation sites from UniProt, including both experimentally verified with publications and computationally predicted annotations. To ensure low redundancy and generalizability, we removed similar proteins based on global sequence alignment and split data into training, validation, and testing. Next, we extracted features from each residue using two complementary sources. The first is probird, a transformer-based protein language model, provided a 1024-dimensional embedding capturing global sequences and contacts and across the whole protein. And the second is the SSP tool. These outputs we use to calculate solvent accessibility and degree of freedom within a 10 angstrom radius. We also included motif-based features, such as the presence of the canonical NX-SOT motif, excluding proline at the X position. So these features were concatenated into a vector for each residue and fed into multi-layer perceptron classifier. This classifier composed of three fully connected layers with ReLU activation and dropout regularization. To ensure robust evaluation, we also tested the model on the NGLY-DE independent benchmark dataset. This dataset contains over 3,000 asparagine residues from glycoproteins, excluding from the training data. It is worth noting that the glyco-DE independent datasets focusing on the sequence window of 20 residue around the asparagine target. We curated a dataset of UniProt annotated glycosylation proteins and mapped them to the ClinVar single nucleotide variants to study the clinical significance and the essentiality of glycosylation as well to address the other study aim. This were further linked to NOMAD to retrieve population-level metrics, including allele count, allele frequency, and filtered allele frequency. For pathogenicity predictions, each missense variant was represented by first 1024-dimensional embeddings from probird, capturing global sequence contact. Second, a 225-dimensional GSNet vector representing local structural and physical chemical properties. For the SGLY, the prediction model, focusing specifically on the asparagine residues, SGLY achieved an accuracy of 95.8 with an F1 score of 0.87. Precision was 0.778, while recall was 0.99. The RCAOC for asparagine residues was 0.975. The SGLY outperformed existing tools such as the AMGLY and deep N-glyprid across all major metrics on the NGLY-DE benchmark dataset. For the essentiality aspect study of this project, in order to investigate the clinical and evolutionary relevance of glycosylation, we compared variant burden across glycosylated and non-glycosylated sites. For glycosylated sites, it had a significantly lower proportion of pathogenic variants of 10% compared to 30% in non-glycosylated regions. This suggests a stronger purifying selection. And for sites with experimentally evidence of publications, it had a higher pathogenic variant rate of 22%. This is likely due to the increased clinical focus and annotation as well. So in the population scale data from NOMAD, the variants at glycosylated residues were enriched in lower frequency beans for both AF raw and FAF95, reinforcing their functional constraint and evolutionary importance. Our pathogenicity classification model integrating the probert and GSNet embeddings achieved 90.91 accuracy and an AUC of 0.957 in classifying pathogenic variants within glycosylation-related proteins. We also conducted a TSNE visualization, which showed the combined embeddings yield clearest clustering. This shows additive values from both representations of probert and GSNet. In summary, we developed SGGly, a high-performance model for unlinked glycosylation prediction that integrates transformer-based sequence features with structural-aware embeddings. Our nomad-wide variance analysis further supports the essential role of glycosylation. This shows reduced pathogenic variations and stronger evolutionary constraint at glycosylated sites. SGGly is freely available at our BioSic Lab web server and can support research in variant interpretation and glycosylation biology as well. Thank you.